Good morning, everybody. My name is Jacob Palmer, and before I begin my presentation, I'd like to thank Dr. Awolowo, who's the lead author of this publication, as well as the faculty advisor for the presentation, and Dr. Joan Davis for the incredible opportunity to represent the class of 2024 at this year's Research Day. We have a limited amount of time to cover today's material, so if you're interested in reading our complete manuscript, the DOI is here on the introductory slide, and you can also find a clickable link on my reference list on Cubify. All right, I believe it's best to start with a bit of background information before I explain what our goal was for this study. Osteosarcoma is the most common primary malignant bone tumor in humans, and in rare cases, primary osteosarcomas can actually develop in the jaw. Several genes are associated with osteosarcoma, namely this SATB2, MDM2, and CDK4. These latter two are both found in the same chromosomal segment, which is the 12q 13 through 15 region. Immunohistochemistry, which I'll refer to as IHC for the remainder of this presentation, is a form of immunostaining that involves the use of an antibody to identify the presence of an antigen in a tissue sample. Previous studies have used IHC to find SATB2 markers to differentiate between osteosarcoma of the long bones from malignant bone tumor mimickers, and they've also used IHC to find MDM2 markers to differentiate low-grade osteosarcomas from other jawbone mimickers. With this background information in mind, the goal of our study was to report the clinicopathologic features of osteosarcoma of the jaw and to determine the value of SATB2 and MDM2 immunohistochemistry in differentiating OSJ from other common jawbone mimickers. For the materials, I want to focus on the samples themselves. We used 24 samples in this study. 11 of these were OSJ, including a single small cell variant, 7 ossifying fibromas, and 5 fibrous dysplasias. These 12 here were benign lesions, and 1 primary Ewing sarcoma of the jaw. All of the OSJs we retrieved showed histologic features of high-grade osteosarcoma, and for our antibodies, we used the EP281 antibody for SATB2 and the IF2 antibody for MDM2. Both of these are produced by Cellmark in Rockland, California. I also want to note that given the nature of our experiments involved in this study, this project was actually exempted from review by the IRB of ATSU. For the methodology, we used fluorescence in situ hybridization, also referred to as FISH, to determine if our samples were positive for the SATB2 or MDM2 oncoproteins. I want to draw your attention here to the criteria applied for positive IHC results in these samples. Only nuclear staining was considered positive in our interpretation, and the percentage of positive tumor cell nuclei was approximated by visually scanning the slides at medium power. Staining of at least 1% of neoplastic cells was considered positive, and the distribution of the staining is as follows here. 0 to less than 1% would be a negative, greater than 1 to 10% is focal, greater than 10 to 100% is diffuse. And then we also semi-quantitatively graded this as weak, moderate, or strong for both the SATB2 and MDM2 oncoproteins. For the results, there's several things I want to point out to you here. All cases of OSJ and all of the benign lesions that I mentioned, those are those fibrous dysplasias and the ossifying fibromas I mentioned in my materials section, they were all reactive for SATB2, so a positive test result. The only thing that tested negative was the Ewing sarcoma of the jaw, and I'll explain more on why that's important in a future slide. As for the MDM2 immunohistochemistry, this was only positive in 3 of the 11 cases of osteosarcoma. So all the benign lesions as well as the Ewing sarcoma, all negative, and then 3 cases of the osteosarcoma were positive. We also, as I mentioned earlier, conducted FISH analysis. This was negative in all interpretable cases, and we only had 5 of the 24 overall cases that were interpretable to begin with, so that's also something that I'll discuss in a future slide. I want to go into a little bit more detail on the SATB2 results we had. As you can see here, and as I mentioned earlier, of all the samples, only the primary Ewing sarcoma of the jaw failed to express any nuclear immunoreactivity. This is an important thing to note, actually, as Ewing sarcoma and small cell variants of osteosarcoma share incredible histological similarities. This makes it pretty difficult, actually, to distinguish between each other under a microscope. As you can see in these figures, there's dark brown chromogen staining that's present in the slides of the small cell variant of OSJ, while no staining is evident in the Ewing sarcoma sample at all. As for the MDM2 status, 3 of the 11 osteosarcoma cases we evaluated were IHC positive for MDM2, while all the benign lesions and the Ewing sarcoma were negative. There are several important things to discuss here, so let's start with the SATB2 IHC. As I said earlier, this was positive in all the OSJ that we evaluated, as well as all the benign lesions, while it was negative in the Ewing sarcoma that we had. While we only had one sample of Ewing sarcoma, other studies have had much larger sample sizes and found that extremely high quantities of osteosarcoma can be positive for SATB2, while the overwhelming majority of Ewing sarcomas will be negative. This leads us to be pretty confident in the reliability of our findings. As for MDM2, only 27% of our samples were positive for this marker, and the literature reports a much wider range of positive results for this. At the present time, we think that this wide range can be the result of various things like tissue fixation, decalcification of samples, antibody cloning and/or dilution, as well as antigen retrieval. All of these things can vary from study to study. Additionally, we discovered while we were writing our discussion section that other studies have achieved much higher positive results using a different antibody for MDM2 known as SMP14. Future studies may wish to use this antibody instead in order to achieve a higher degree of sensitivity to MDM2. A little bit more on the MDM2 amplification significance. While not all of our samples tested positive, the presence of MDM2 amplification presents a therapeutic opportunity. At the present time, MDM2 inhibitors have been developed to suppress proliferation and promote apoptosis in both osteosarcoma and other cancers. And while these chemotherapeutic agents are not fully successful on their own, when you use them in combination with other therapeutic agents, you can drastically increase the success rate of treatment for malignancies that test positive for MDM2. There are a few important limitations I want to point out here. As I mentioned at the beginning of this presentation, CDK4 is another important gene that exists in that same region as MDM2. We didn't evaluate markers for CDK4 in this study, but doing so may have given us a better representation of the amplification of that chromosomal region in our OSJ samples. This is especially important to consider as some studies have shown that overexpression of MDM2 and CDK4 is actually mutually exclusive. I also want to point out some noteworthy findings. When it comes to SATB2, we determined that this cannot be used to differentiate OSJ from benign fibro-osseous lesions, but it could serve as a valuable diagnostic marker to differentiate small cell variants of OSJ and Ewing sarcoma of the jaw from one another. As for MDM2, we found that this marker has some potential, specifically when it comes to differentiating low-grade or high-grade transformed osteosarcoma from benign fibro-osseous lesions. While we're on this subject, another thing I want to bring up is that some cases of OSJ expressed MDM2 while others did not. This may indicate that the 3 high-grade osteosarcoma cases that we evaluated that were positive for MDM2 may have undergone transformation from a low-grade osteosarcoma. Last of all, I want to make it clear that the absence of MDM2 immunostaining shouldn't be used to conclude that a lesion is not an osteosarcoma because we had those mixed results. All right, this concludes the presentation. As I mentioned at the very start, the results of our study have already been published online, and in fact, our manuscript was selected to be highlighted on the front page of the publishing journal. To my knowledge, this is the first study that tested for the presence of both SATB2 and MDM2 in the same sample lesions at the time of publication, at least. And I want to thank all my co-authors for the work that they put into publishing this article. I also want to give a special thanks to Drs. Awolowo, Summersgill, and Davis for giving me the opportunity to present this paper to you today. Thank you all for your time and attention, and I believe we have some time for questions.